The further steps encompasses the various fates of pyruvate. Two important fates of pyruvate are reduction and oxidative decarboxylation. Reduction of pyruvate takes place in cells without mitochondria or anaerobic conditions in cells with mitochondria. Similarly, oxidative decarboxylation of pyruvate takes place in aerobic conditions in cells with mitochondria. Now, the reduction of pyruvate takes place in cells without mitochondria. So, if any cells do not have mitochondria, there is no question of the reactions taking place inside the mitochondria, such as TCA cycle or the various steps of electron transport chain followed by oxidative phosphorylation. So, in the absence of all these things, the pyruvate is reduced and does not enter into any of these steps of oxidative phosphorylation. Similarly, in anaerobic conditions in cells, even with mitochondria, if the oxygen is relatively deficient in cells with mitochondria, such as exercising skeletal muscle cells, the pyruvate, instead of entering into the oxidative phosphorylation, TCA cycle followed by electron transport chain followed by oxidative phosphorylation, it undergoes reduction. Similarly, oxidative decarboxylation is the conversion of pyruvate to acetyl-CoA, which is an important substrate for TCA cycle. Takes place in aerobic condition in cells with mitochondria, and since TCA cycle takes place in the mitochondrial matrix, the conversion of or the oxidative decarboxylation of pyruvate to acetyl-CoA is an important step before the TCA cycle takes place in the mitochondria to generate very important substrates or compounds for the successful running of electron transport chain and the subsequent oxidative phosphorylation for generation of ATP. Now, reduction of pyruvate. The enzyme that catalyzes the reduction of pyruvate is lactate dehydrogenase, and pyruvate is reduced to lactate. There is a reversible reduction of pyruvate to lactate. So, along with the reduction of pyruvate to lactate, there is an oxidation of NADH to NAD+. This NADH, which is oxidized to NAD+, is the same NADH which is generated during the oxidation of glyceraldehyde 3-phosphate. Now, once NADH is oxidized to NAD+, in the along with the reduction of pyruvate to lactate, the NAD+ will again be utilized in the oxidation of glyceraldehyde 3-phosphate to generate NADH. And the same NADH will again be used in this step to form NAD+. Now, the conversion or oxidation of the NADH to NAD+ and subsequent reduction of NAD+ to NADH to again get oxidized to NAD+ takes place continuously, and this ensures the continuation of anaerobic glycolysis. Whereas in aerobic glycolysis, this NADH is in fact utilized to provide energy, and it donates electrons to the electron transport chain inside the mitochondria, or to be more precise, in the inner membrane of the mitochondria, and the electron transport chain operates and is followed by oxidative phosphorylation to form ATP. But in the anaerobic glycolysis, this NADH is utilized to continue the anaerobic glycolysis. The reaction, pyruvate is reversibly converted to lactate, or pyruvate is reversibly reduced to lactate. So, along with this reduction, there is oxidation of NADH to NAD+. This reaction is reversible, and the enzyme that catalyzes this reaction is lactate dehydrogenase. The tissues where there is relatively anaerobic condition, or there is lack of mitochondria, and therefore the pyruvate is reduced to lactate in the presence of enzyme lactate dehydrogenase, are lens and cornea of the eye, kidney medulla and testes, leukocytes and RBCs, and exercising skeletal muscle cells. The red blood cells do not have a mitochondria, and the glycolysis is always anaerobic. So the end product of glycolysis in the RBCs or red blood cells are always lactate. The pyruvate is further reduced to lactate.